which is the on the germination of the oocysts to hollow the cylindrical structure with the conical tip, which is enclosed with the shoot apex and first foliage leaves are there. This structure is the coleoptile. The substances is present at the tip of the coleoptile. When the tip is removed, this is no curvature, but they are placed back on the diptation stump with a layer of the relation between the cut surface and curvature is brought. This shows the substances diffuse from the tip to the water soluble, thus moving through the gelation on the region of the curvature. When the tip is cut to the dark and when cut into the placed along with the one side of the dipicated stump. On the side opposite shows curvature, thus the showing the curvature is brought about due to the uneven distribution of the growth promoting substances. The side of the seedlings receiving more grows faster than the opposite side, so thus the resulting the curvature. The substance is responsible for the curvature was isolated in 1928. That is the F.W. Wendt, is a Dutch botanist. He was cut off the coleoptile and replaced them on the agar block for the 1 hour. When this agar block was replaced on the stump and curvature is occurred, that shows that the substances from the tip flowers downwards into the agar block. So this technique was employed for the extraction and estimation of the hormone. The chemical nature of the hormones was identified and named as a indole acetic acid (IAA). As the oxins are soluble in water, alcohol, chloroform, ether, the extraction is done by the solvent method. Okay. So the IAA is universally found to be plant and synthesized from the precursor of the molecule tryptophan found in the leaves and other part of the plants. The tryptophan is converted into indole pyruvic acid, which is further gives rise to indole acetaldehyde. This is further oxidized to form the indole acetic acid. So these IAA is important natural occurring oxygens in plants. The oxygen-like substances synthesized in the laboratory called as synthetic oxygens. So some important synthetic oxygens are indole butyric acid, naphthalene acetic acid, ABA or naphthaloacetic acid, NAA, and 2,4-D, 2,4-dichlorophenoxyacetic acid, 2,4-D. So what is the distribution of the higher class? The movement of the oxygens in plants. The oxygens act at a distance from their source. They move in the basipetal condition towards the morphological base and exhibits the distinct polarity. So the amount of the oxygens in the tissues of the plant must be regulated for the proper functioning and the growth controlling substances. And this is brought about the various ways, that is the oxygen may be bound to be in the protein and thus inactivated. The formation of the inactive derivatives by reaching the with other chemicals can be oxidized since all the IAA oxidases. These enzymes are found in many tissues and brought the important in the regulation of the oxygen level. So some physiological functions of the oxygens. What are the physiological oxygens are there? These physiological oxygens means apical dormancy. They are influenced the phototropism, geotropism, and apical dormancy, parthenocarpy also. So first, the effect is the apical dormancy. In many kind of the plants, terminal buds is the dominant and inhibiting the growth of the buds, lateral branches or buds. So this phenomenon is called as the bud inhibitor is the demonstrated strictly in the potato tuber. So another aspect is the rooting. Stem cutting when they applied at the base of the auxins, this promote the root formation. And generally, IBA indole acid is used for the root inhibition. It has commercial application in the propagation of the economically important plants. Another function is the parthenocarpy. The process of development of fruit without fertilization is called as the parthenocarpy. So without fertilization means there is no seeds, only the fish is present. The development of the fruits in the various ovaries of the flowers and possibly also demonstrated of the ovules into the mature seeds after fertilization, which is controlled. This was first ovaries. They are developing the fruit which is the had no seeds because there was no fertilization of the ovule. The ovary that develop into the parthenocarpic fruit. Further studies have shown that the auxins and related growth substances when applied to the ovaries of the flowers cause the development of the seedless fruit. This technique is of the commercial value for the large scale production of the seedless fruits. For example, grapes, lemon, oranges. These are the involved in the parthenocarpy. Growth movement. The plants may react with the growth movement to directional stimuli, particularly light and gravity. These movement are to irreversible changes resulting from the growth and relating to the direction of stimulus. Such movements are termed as the tropic movements. So tropic movements of the plant's organs are always directive. The direction of the movement determined by the direction from the stimulus to apply the organ move there towards the source of the stimulus. So another application is the geotropism. The movement of the plant organ in response to the force of the gravity is called geotropism. Primary roots grows into the soil length in the lies of the gravity and they are responsible to geotrophic, whereas the stems grows away from the center of the gravity known as the negatively geotrophic. The secondary roots and branch and leaves of the stem are placed horizontally. They are the plesiotropic or diageotropic condition. The response is to gravity. The response in gravity uh, is inductive and can be separated in time. This shows that two separate processes are involved. The perception and reaction. According to certain physiologists, or the it can be experimentally found in the FW vent. And phototropism. Another one is the phototropism. To understanding the mechanism of phototropism to react, it goes back to vent experiments to lead the discovery of oxygen. The movement of the plant organs as determined by the direction of the incidence of rays called phototropism. The stems grows toward some source of light and known as the possibly phototropism. The leaves and stem bend toward the light and known as the Travels are phototropism or diaphototropic. Phototrophism can be easily demonstrated by using the phototropic chamber is there. So another is the sex determination is also. So application of oxygen to increase the formation of the stipulated flowers in the cucurbitaceae. So means female flowers are more increased, fistulated flowers. Another one is the gibberellins. Another growth hormone is the gibberellin. So gibberellins strongly promote the plant growth, especially of the stems. They also have the sticking effects on the flowering and dormancy, and where the first discovery by Japanese scientists. So the Gibberella fusicoria leads to the excessive elongation and internodes to lead to the discovery of gibberellin. The plants infected by this fungus grew gigantic and hard and drawn out the yellowish appearance, were due to the excretion of the certain substances by the fungal. So this is characters as called gibberellin. So biosynthesis gibberellin means there are some names as GA1, GA2, GA3. So some physiological factors of the gibberellins to remove the genetic dwarfism. So is already pointed in the extensive stem elongation in the infected rice plants, which is the the hyperlongation of the stem remains on the sticking influence of the gibberellins on the plant growth when the dwarf mutant of the peas and maize are separated, spread, and the gibberellin. The internodes elongated on the plants assume to appearance normal tall varieties. The tall varieties of the peas and maize do not respond to the gibberellins. The darkness in the mutant is due to the gene mutation is leading to the block in the synthesis of gibberellins at the site of the action. Exogenous application of the gibberellins remove this sort of the dwarfism, bloating and flowering. In the rosette plants such as mustard, cabbage, and radish, the leaves development is profuse and the internodal elongation is retarded. However, just before the flowering stage, this is the dramatic increase in the stem elongation due to the internodal growth. This sudden and rapid increase in the stem growth is known as the blotting. In blotting, the plant's initial rosette growth is due to the poor gibberellin content in the plants that result in less stem elongation. So just before the blotting, there is increase in the gibberellin biosynthesis, which leads to blotting and flowering. Not only in the rosette plants, but in majority of plants, the gibberellins induce the formation of the flowering primordia and enhance the flowering. Another application is the seed germination. So gibberellins are regarded as the patent and germination promoting the hormones. During this early process, the seed germination gibberellins which is synthesized in embryo. So they facilitate the hydrolyze of reserve food material in the storage parts such as endosperm and their movement to embryonic axis. This sort of the mobilization of reserve food material which helps in the germination of the seeds. Parthenocarpy. So in grapes and several other plants, there is a gibberellins induced parthenocarpic fruit, and sex determination is also the application of gibberellins to increase the formation of the male plants in cucurbitaceae. Another growth hormone is the cytokinin. The Heberland discovered the known substances present in the vascular tissue of the various plant-stimulated cell division in the cut into potato tissue. So this is the cytokinins are there, some physiological functions are there. The cell division. So as already mentioned, the cell division is the main function of this group of these substances. It was this activity that lead to their discovery and the promotion of the cell division remains a prime requirement of the substance classified as a cytokinin. Another application is the cell elongation. Although cytokinins are mainly concerned with the cell division, they are also capable of the enhance the cell elongation, particularly in the leaf disc and cotyledon. Another application is the morphogenesis or organogenesis. Cytokinins, by interacting with the auxins, regulate the process of morphogenesis in plant. In tissue culture media, highly cytokinins to auxins ratio stimulate the shoot differentiation. It is called as a coleogenesis also. On the other hand, low cytokinin to auxins ratio it induces the root formation, which is called as a rhizogenesis. Depending on the concentration of the cytokinins, they are developed as the coleogenesis or rhizogenesis. Delay in another application, delay in the senescence. The aging process in plants known as the senescence. In leaves, such an aging process result in the yellowish of leaves. The yellowish of the leaves is mainly due to the breakdown of the chlorophyll, nucleic acid, and proteins. Cytokinin delay the process of senescence in leaves and other organs and also in the entire plant. If cytokinin solution is applied to yellowing the leaf, it is turned black to green color. The ability of the cytokinins to delay the process of senescence is known as a Richman-Long effect is called. Another one is the stomatal opening is also. So cytokinins to induce the stomatal opening by the enhance the movement of the potassium ions into the guard cells from the surrounding epidermal cells. So another growth inhibitor, that is the abscisic acid, ABA. ABA we call as a growth inhibitor. So abscisic acid is an important growth inhibitor substances in plants. The studies on conducting by the scientists, that is the organ and dormants brought into the light are the existence of abscisic acid in plant kingdom. In 1963, the Adicott and these Hawkers discovered these two substances. Cotton fruit is responsible for the abscisian of fruit. They are named as the abscisian 1 and other one abscisian 2. They are named. So the abscisic acid is the 15 carbon terpenate. So abscisic acid is widely distributed in the plant kingdom. It is present in all the vascular plants, several mosses, some green algae, and some fungi also. So the next one is some physical function. Physiological functions are the abscisian. They remove the important function is attributed to abscisic acid. During the discovery, it was involved in the abscisian of the various organs such as leaf, flower, and fruit also. It has been suggested that the abscisic acid is accumulated in the abscisian zone and finally leads to the detached of the organ by weakening of that area. So further, It is also established in the fact that ethylene, another phytohormone, plays important regulator in the abscisium. Dormancy is another application. The dormancy, which is earlier studies attempt to establish the link between abscisic acid concentration and dormancy. While some studies have been reported to correlation between the ABA concentration and the intensity of dormancy, in several other cases, the results are not connected. This is no substantial evidences and the ABA is the primary factor in the inducing dormancy. So another one is the stomatal movement. In 1969, 1969, the Bright reported 40-fold increase in the ABA content in the wheat leaves when the subject to water stress. So the studies clearly reveal that when the water level of the plant will be decreases, the ABA biosynthesis increased. The ABA in turn induced the stomatal closure, thereby the lower water loss due to the transpiration. In this manner, the ABA acts as a natural anti-transpirant in the plants. It helps to conversion of the water during the drought stress conditions. Another one is the seed germination. During the maturation of the embryo in seed, ABA accumulate in it is a large quantities. This helps to prevent the germination of the seed before it is released from the fruit. In dry seeds, the level of the ABA are high and act as a potent germination inhibitor. Formation of the penetrating buds. In lemna, the several other aquatic plants, the abscisic acid induce the formation of the penetrating buds when the ponds and dry up. These buds develop into the plant when their water is availability. This is improved. Another inhibitor is ethylene. So ability of certain gases to stimulate the flowering has been known as for many years. In 1910, the Cassin suggested that organs should be not be stored along with the banana during the transportation. In some imitation from the organs causes bananas to ripen the prematurely. So ethylene is the simple carbohydrate with formulation C2H4. The ethylene is the unique plant hormone in the gaseous nature. Ethylene is frequently produced when the high concentration of the oxygen is supplied to the plant tissue. So here some commercial pineapples grows imply the ethylene is the ethylene is replacing the substance to increase the flowering. So flowering senescence, ethylene to accelerate the process of senescence in flower. Wound healing also. Ethylene stimulates the cell division and helps in the process of healing of wounds. So that's why the ethylene is called as the whole wound healing hormone. So other functions also there, that is the induction of the root hair formation. So increase the production of the pistillated flower. Assignments was the Kumar sir. Final year. So wait some time. Kumar sir is in the class. Okay, you hand out to the Kumar sir. Wait some time. So in the pistillated flowers are to develop. And another one is the acceleration of the abscission of the leaf, the flowers and fruits also. So it reduces the apical dormancy by reverting the effect of the oxygen. So induction of the parenchyma in aquatic plants also. So here some physiological functions also there. Fruit ripening is the main function is stimulated by the ethylene. In fact, the ethylene is often being referred to as the fruit ripening hormone in a variety of the fruits, such as the bananas, apples, and mangoes. This ethylene is released in turn to acceleration to process the fruit ripening. The exogenous application of the ethylene to unrip fruit is also acceleration, the process of ripening. So another one, the triple response growth. The triple response growth is means three pattern growth, that is the etiolated pea seedling. These three pattern growth are the inhibition of the elongation, stimulation of the lateral growth, and induction of the trans to geotropism. This is the triple response growth we call. Another one is the flowering. The several plants, ethylene is suppressed delaying the flowering. However, the ethylene is biases that have been developed to make use for this aspect of their activity. It is also autogenous action of the acetic acid. Some role of the growth hormones in the different field. So in agriculture field, growth hormones are how used in the agriculture field. The numerous developmental response of the plants are under auxins control. Some of these response can be also be produced by the use of the auxins preparation, which may be rapidly from the outside as a growth regulation regulators. These are synthesized auxins are there. Though the synthetic auxins do not possess a wide ranges of activity as the IA, they will do show their effect on the growth and development of the plants similar to natural auxins. Some synthetic auxins is recognized after the identification of the natural auxins. They are the indole butyric acid and naphthalostic acid. They are replaced with the natural auxins. So synthetic auxins have mainly an important particular use in the orchids. NAA and 2,4-D are employed to increase the fruit and fruit size. They are also used to prevent the pre-harvesting drops to thin cut of the supplies fruit by inducing the fruit drop. Another application is the rooting. Stem cutting, when they applied to the base of the auxins, they promote the root formation. Generally, the IBA, indole butyric acid, is used for the root initiation. It has commercial application in the propagation of the economically imported plants. Weedciders, 2,4-D and 2,4,5-T. 2,4-D, 2,4-D dichlorphenoxic acid and 2,4,5 trichlorphenoxic acid. They are the selective weed ciders, weed killers, nothing but weed ciders and weed killers. So phenoxyestic acid, which is selectively eradicated dicot weeds, is a very good value for to the farmers within they are eradicating the dicot weeds, growing the amidst their monocots crop by spraying the 2,4-D and 2,4,5-T. Another application is the apical dormancy also. In many kinds of the plants are terminated but in the dominant, inhibiting the growth of the lateral buds. This phenomenon is called as a bud inhibitor. So another one, the breaking dormancy. So by spraying the gibberellins of the dormants of the buds, it is broken and allowed to sprout. To another one, the fruit and flower and fruit formation. The flowering of the plants appear to be released to the hormone activity. The formation of the concentration, this is merged since it is not been possible to isolate the flowering hormones. So gibberellin induced the flowering in several plants. Some crops where the flower are not needed, the formation can be keep checked by the spraying the chlorophenoxy pronionic acid by spraying the 2,4-D. It is easy to fall. The fruits can be checked. This can also spraying the auxins to get a large size of fruits. Abscissin. The abscissin of the leaves, flowers, and fruits was in the past controlled by the auxins. The abscissin of the leaves and fruits is due to the decrease the oxygen supply in them to control the compared to the stem. When indolestic acid, there is a concentration in the leaf, concentration in the leaf, and fruit is higher concentrated than stem. So abscissin is normally does not occur in this when they are growing the, but when maturity time the IAA is concentration decrease and abscissin is occur. This decrease in the oxygen level in the case of the is usually accompanied by increase the level of the abscisic acid. The fruit grows make the particular use in these relationship. In the spring, they spray the auxins on stems, which increase the hormones concentration of the stems, the resulting the premature abscess in the fruit. Another application is the parthenocarpy. This is already discussed that one. Another one, the seed germination. And cytokinins and gibberellic acid helps in the germination of tobacco and lettuce seeds. They are also checked the apical dominant and the activity of development lateral buds also. So another topic is the photoperiodism. So photoperiodism means the seasonal changes in day length and condition. Conditions performed in the influence of the process of the flowering in many plants. This is the significance of the light in flowering. It was first demonstrated by the Jarner and Allard. These two American breeders, plant breeders, they are described in 1920. They found that the newly developed variety of the tobacco, Maryland mammoth, grew vigorously throughout the summer but did not flower. This is in the contrast to widely grown variety of Maryland narrow leaf, which the flowers in summer. After through the investigation, the Journer and Allard is observed the shortening of the daylight period, induce the flowering to Maryland, Mamanchi even this summer. So subsequently, this was observed in many plants. The day length is crucial for the inhibition of the flowering, and this phenomenon has come back to known as the phototropism. The photoperiodism can be defined as the flowering response of the plant to the relative length of the daylight and dark period. These plants in which is the flowering is readily induced by the exposed to the short days are known as the short day plants. Similarly, those plants in the flowering in the favored by the long days, they are referred as the long day plants. So, however, some days, some plants are not under the control of the daylight conditions. Such plants are the day natural plants. So, short day plants is example. That is the, as I mentioned, the shorter days induced flowering in the short day plants. Short day plants are divided into here. Two types are there. qualitative short day types and quantitative short day plants. These plants have obligatory requirement for the shorter days. If the plants is fail to get the exposure to the shorter days, they remain the vegetative throughout their life without flowering. Xanthium, for example, xanthium, chrysanthemum, soybean. These are the qualitative short day plants. And another one is the quantitative short day plant. In these plants, the flowering is readily to induce due to exposure to the short day. If these plants is failure to get exposed to the short days, they will not remain vegetative but ultimately flower. But the flowering is delayed and poor. Another one is the long day plants. These long day plants is require more duration of the light period for the flowering. Similarly, they are the short day plants. Long day plants can also divide into two types are there. Qualitative long day plants. These plants is obligatory requirement for the longer days for the flowering. Example, radish and henna bean. Qualitative long day plants. These plants is require the longer days for the flowering. Initially is not very obligative. These plants. So these plants flowering the profusely if they are getting the expanse to the longer days. For example, peas and wheat is also. So another topic is the phytochrome. Okay. So the morphological process in plants which are influenced by the light is known as photomorphism. The flowering and seed germination are two important to photomorphogenetic process, a major advance in our understanding the photomorphogenesis. So, for example, in the lettuce, the studies on the germinate the implied in the red and infrared lights lead to discovery the pigment known as the phytochrome. So, this phytochrome is the conjugated protein is consists of the protein and non-protein. The chromatophores is the physiological activity of the part is involved in the observation of the light. The phytochrome influences all the phytomorphogenetic process. So here, short day plants and long day plants are there. In the short day plants, they are the dominance in PR means red absorbing plants are there. P is 380 and far red observing plants P 735. These are there far red observation. Another topic is the vernalization. So before flowering, many plants require the period of cold treatment. Some cereals are required this cold treatment. Wheat is very good example. Some of these varieties of the wheat is controlled the spring wheat. So these are usually planted in the spring and they are flower in the same season. So some varieties which are the known as winter wheat should be planted only in the up. These plants flower and fruit after winter season. They are the cold temperature of the winter are essential for the winter wheat. Without this cold treatment, these plants are not flowered and flowering was reduced. The So this process was conversion of the winter varieties to summer varieties by the cold treatment called vernalization. In several plants, such as the cereals, that is the Secale cereal, is exposed to the seeds to cold treatment will the sufficient. Another Iscavim niger is to be vernalized when the plant is 10 days old. In some plant is vernalization can be effected only after some vegetative growth has takes place. So it has been observed that mainly keeping the apex of the region which is perceive of the effect of the vernalization. There are some expansion of this one. For example, Walensky in 1961 to 62 has suspected the vernalization in the leaves and root of the Lumeria vacuinis. The mechanism vernalization is a complex process. It is involved several processes are there. That is the Michels in 1962 is postulated the substance called vernalization is responsible for transmitting the vernalization stimuli. So, Langaf United States found in the application of the gibberellin would be replaced the vernalization in many cold requirement plants. However, the gibberellin is not a vernalization. Vernalization because it response the plant to gibberellin is different from cold treatment. So another in 1968, the Thalikian is explained the mechanism along the plants of the flowering phase. He suggested that these plants under low temperature vanillin is produced. This along with the another substance called hen, which is present in the long day plant to cause the flowering in the long days. In short days, the vanillin is not converted to gibberellin, and so flowering does not occur. So this is known as vernalization. So it is more likely to some hormones and growth substances involved in the flowering induction. How they are related to the process of vernalization is some plants. Okay.